some macrophages are called dendritic cells. They have a very special function. These generally macrophages are out there engulfing bacteria and foreign material, invaders and stuff like that. Dendritic cells are doing so as well, but they're doing so as a collection of stuff to present to T cells later on in the lymphatic system. These guys, their main goal is to fight off invaders, engulf them, eat them, get rid of them. These guys are sampling what's coming in. So that sample of bacteria, parts, and debris and stuff can then be presented to lymphocytes so that we can establish memory of the things that we're being exposed to. So dendritic cells are out there picking up material that they will bring to present to lymphocytes.
So is it like a B cell?
It's not like a B cell. And we'll get there, but no, it's not like a B cell. Although B cells do that. The other thing that we can say about these two cells here is that they are also antigen-presenting cells. So back to your question, Laura, B cells also are antigen-presenting, and I'll say more about that when we get to chapter 16. Well, yeah, we're chapter 16. These guys are antigen-presenting. What that means then is that they can engulf bacteria, engulf parts of bacteria, and then present those parts to T cells. And in presenting those parts to T cells, they're asking T cells, hey, what do you think about this bacterium I've got? T cells will serve then to regulate, meaning that it will decide, hey, that's a bad bacterium, we need to full-blown immune response. Or it might say, ignore that guy, he's not going to bother us. So the T cells will serve to regulate what's going on with the immune system based on the antigens that are being presented to it. So these two cells can present antigens, and we'll see here shortly that B cells also can present antigens. Brings us to our third type of white blood cell. The lymphocytes. The lymphocytes are mainly involved in the adaptive immune response. So we're going to talk about these in much more detail in chapter 16. Their role in the innate immunity really is minimal at best. The cells of the lymphatic system show molecular specificity. So those cells then will only bind to specific molecules. So B cells, certain B cells will only bind to certain molecules. Certain T cells will only bind to certain molecules. Very specific recognition and attachment. Two types of lymphocytes, B cells. And B cells are antibody producers. They're the only cells that produce antibodies. And then T cells are the other type of lymphocyte. T cells do not produce antibodies. They do, there are different species of T cells. They can be either cytotoxic, and we've talked about what that means. So some T cells destroy infected somatic cells or destroy cancer cells. And other T cells are helper T cells. These actually are the most important T cells, if you will, of the immune system because they regulate what the B cells do, whether the B cells are going to be producing antibodies or not. Turns out it's the AIDS, or it's the T helper cell that the AIDS virus targets. And so as you wipe out this population of white blood cells, you depress your immune system's ability to respond to infection. A lot of times it's not the virus, the AIDS virus itself, that causes the death of AIDS patients, but it's opportunistic infections that cause the death simply because the body cannot respond with an appropriate immune response. One other type of lymphocyte natural killer cells. Natural killer cells seek out, destroy cells that are abnormal. So again, cancerous cells or cells that have been invaded. These guys, and we'll talk more about this, rely on what is called antibody-dependent cellular cytotoxicity, meaning that they're only going to target cells that have been bound by antibodies. Again, we'll say more about that here in chapter 16. Antibody-dependent cellular cytotoxicity. Antibody-dependent cellular cytotoxicity, and you don't have to worry about it until chapter 16. And then they spell it out for you in chapter 16.
Mr. Hayes, do you also call natural killer cell cytotoxicity?
Yes, I would. Cellular cytotoxicity. So, one of the things that needs to happen for the immune system to work correctly, for them to communicate back and forth, is just that, cell communication. The immune cells have to be able to communicate with each other to know what response to implement. And so when we look at the types of communication we see, right away we start with the cell membrane or the surface where we see receptors. Membrane proteins that allow for the cells to communicate with each other. So these proteins on the surface, and so if you imagine here, say, cell, we're talking about, you know, specific proteins that are embedded on the surface, right here. And specific molecules will bind to these surface receptors. And in that way, the cell can respond to the types of surface receptors that are being bound. Now, imagine if you've got specific surface receptors for a type of microbe or a molecule that a microbe might have. When those become bound, it can trigger you to release a chemical, a messenger, a signal that alarms other cells, maybe draws neutrophils to the area, maybe brings macrophages to the area. These surface receptors really allow for the cell to interpret its microenvironment, to know what's going on around the cell. How do the other cells communicate with it then? By releasing chemicals into its microenvironment. If you want to talk to another cell, you release chemicals that that cell can hear, or that cell can be bound by. Okay? If you're releasing chemicals and they're not binding to any surface receptors here, then this cell is ignoring that message that you're sending, and won't listen to it. So surface receptors will serve as a form of cell communication. Something that binds to a surface receptor is a ligand. So we can refer to a molecule that is binding to a specific receptor as a ligand for that molecule or that receptor.
Can glycoprotein be a surface receptor?
Sure it can, actually. Absolutely. Glycoprotein, glycolipids not so much. They serve more as an identifier. Some things can attach to them, absolutely. And we should recognize that we can change the surface receptors as well.
Are they also specific?
What do you mean they?
Well, the surface receptors of proteins that are there, do they only bind the ligand that binds them, or are they specific then?
Exactly. So this surface receptor here will only bind to one specific ligand. This surface receptor will bind to one specific ligand. And so yeah, the surface receptor is very specific. So our cells can change these surface receptors depending on, the messages that they're receiving from the microenvironment, but so can bacteria. Okay? So bacteria can change their surface receptors as well, or their glycoproteins, change the molecules that they have on the surface. And so that can change whether or not they will be bound by, say, a macrophage. Say if a macrophage or an antibody recognized a specific molecule, and then that bacterium changed that molecule, then that antibody is not going to bind to it anymore. The macrophage isn't going to be able to recognize it. So we can change our surface receptors, and so can bacteria. And so it's one of their ways of getting around our defense mechanisms. Cytokines. Cytokines are proteins that the cells secrete. So these are things that the cells are going to release, and they generally are going to induce the cell that they're binding with. So you could have, let's say, a T cell here, and it might release a bunch of cytokines, and you have over here a macrophage, and so those cytokines would bind to this macrophage and then induce this macrophage to do something. Maybe it causes the macrophage to grow. Maybe it causes it to die. Maybe there'll be movement. Some kind of differentiation might occur. So cytokines serve to deliver a message and induce a behavior from the cell. Cytokines can act locally, so that they just affect the cells in that particular area. Maybe they are regional, or they can be systematic too. So they can be throughout the body as well. The key to their ability to work is that the cells that they are targeting must have a receptor for that particular cytokine that's being released. So the cytokine will only affect those cells that have receptors for it. Adhesion molecules. These are molecules that allow cells to adhere to other cells. You'll find that the endothelial cells that are lining our capillaries have adhesion molecules that will bind to monocytes. So if you've got an infection going on in a tissue, these adhesion molecules can bind to those monocytes as they're flowing past, and so it slows down the monocyte to a tumble and then a stop, and then the monocyte will move into the tissue. Okay, so that's what I have for you tonight. Let's give these exams back.